Okay, let's make a start. Hi everybody, thank you all for coming. First of all, I'm just going to do a brief bit of housekeeping. So this is the last CNI of the year, so thank you to everyone who has attended regularly, and thank you to all of the speakers who contributed. I'd like to encourage people to come forward to present next year. This can be short science presentations, of works in progress. It doesn't have to be a polished piece of work for you to present. It can be a methods talk, and you don't have to be the world's leading expert on a method in order to give a talk. Obviously, if you are, that's great, but giving a talk on it might help you to flesh out some of the details and become more of an expert. And of course, we always want to hear about the research that people are doing. So any research talks that people want to give, that's also very welcome. The dates are going to be from the 26th of January to the 30th of March. So please do get in touch if you want to give a talk or you know anyone who does. It's my great pleasure to introduce Lucy Summers from the Sussex Colour Group. She is in the final year of her PhD, she's going to be telling us about a fascinating and slightly less well-known EEG method, SSVEP, and its application to another fascinating topic, which is colour vision deficiencies. So over to you, Lucy.
Hey, hello. Thank you for coming. So yeah, as Chris summarised, I'm going to be introducing the steady state method, its features, components, constraints, how it's being used by other researchers, and how I'm using it to identify neural gain control in the colour vision system. So in its most basic terms, an SSVEP is an amplitude collected in response to a flashing stimulus, such as this. I can play this slide, which I can't. So this disc would be flashing at a particular frequency, and this is what you look for in your data. So normally with EEG research, you present a stimulus to your observer, and then you watch the brain respond as time goes on. And then you allow the brain to return to a resting state before you show the next stimulus. Now, this differs with steady state stimulation, where you present the stimulus over and over and over again, rapidly, specifically so that the visual system doesn't get a chance to return to this resting state. Now, what this does is it entrains the neuronal population to respond in synchrony, which means that the brain activity that you then observe is repeating at the same frequency as that of the stimulus. So where with ERPs, you take your data and average across all of the trials and all of your observers to obtain the waveform. With steady state stimulation, you take the raw EEG data and you perform a Fourier transform. Now, this is a mathematical process that decomposes the messy EEG data into all of its constituent sine waves to see at what frequencies activity is repeating at. And what this results in is a plot somewhat like this, where you have the frequency increasing along the x-axis and amplitude of brain response on the y-axis. And you look for specific peaks at certain frequencies. So one of the first features of the SSVEP is going to be a narrow peak at the frequency your stimulus is repeating at. And you also have harmonics of this. So this is a peak of activity at integer multiples of your frequency stimulus. So some of the constraints to be aware of when you're running an SSVEP study is that it's going to be very important to pick the right speed for your stimulus. basically you need your stimulus to be fast enough that the brain doesn't have a chance to return to the resting state before you show the next stimulus, which this is because this creates the synchronized and frequency tagged response that you're going to look for. So if your ERP response function usually ends after 400 milliseconds, you know that you need your frequency of stimulation to be faster than 2.5 hertz. Alternatively, you also need your stimulus to be slow enough that the information has time to reach your region of interest. So here we have the visual pathway. Now, if you're interested in low-level sensory processing, you know that you want the information to have time to reach the V1. So you need to repeat the stimulus no faster than every 40 milliseconds. But then to reach the V2, it needs about 50 to 70 milliseconds. But if you're interested in higher level processing, like face processing, it needs to get all the way to the V4 or the IT. It's going to take longer. So for low level perception, you want a stimulus that's slower than 25 hertz. But if you're interested in high level processes, it needs to be slower than 12.5 hertz. Now, one of the key exciting things about SSVEPs is the incredible signal-to-noise ratio you can get, but you do still have to be careful to avoid areas of noise in the system. So you have endogenous oscillations. So here you can see at 10 hertz, you have a peak of the alpha wave in the brain. You also have to deal with a lot of noise at low frequencies. So this is going to be caused by any interference that happens slowly, any change across the testing protocol. And you also have to be aware of 50 hertz electrical noise, which is going to give you a precise peak at 50 hertz. So as I mentioned, with SSVEP, the signal-to-noise ratio you can get within a short testing frame is very exciting. So here you can see after a short testing session, I was able to get signal-to-noise ratios of 15, 20 and higher. And you can see these peaks here. So there are also criticisms that because SSVEPs require very fast flicker, that it's always going to elicit a sensory rather than a cognitive response. However, there is evidence of higher level interactions. So you have research that is picking out impacts of fatigue, but also of attention on SSVEP amplitudes. And SSVEPs have also been used to identify face processing effects. So here, Rossian uses SSVEPs to identify the difference between face processing and individual identity recognition. So here you can see one run of stimuli featuring different faces elicits one amplitude with a different amplitude when the same face is presented over and over again, representing a sort of recognition suppression. And they manipulated this further. So by inverting the faces, they found this removed the effect of recognition. And then there was no difference between the amplitudes of the two conditions. So you can also use a number of different paradigms. So the oddball paradigm is very useful for higher level processing. Here in design 1, you can see they are repeating, they are changing the stimulus at one frequency, for example, perhaps 9 hertz, and then presenting a face at a separate frequency here every 3 hertz. So you'll be able to see once you perform a Fourier analysis, a separate peak for the low-level processing and the higher-level face processing. Now in design 2, you can see a similar sort of design to the Russian from the previous slide. Here presenting the same face each time at one frequency with different faces at a quicker frequency, allowing you to identify amplitudes in response to both face processing and recognition within the same trial, testing them simultaneously. So you also have the sweep. So a sweep is a paradigm where you are varying one dimension of the stimulus in multiple different ways, in changing them by increments so that you can rapidly categorize the response to one dimension of your stimulus. So this is often used in preliminary studies to help you find the right characteristics, constraints, and perhaps the right frequency to test your stimulus at. Now, SSVEPs also allow you to test multiple stimuli within one trial. So here we have a checkerboard in which all of the red checks are repeating at one frequency and all of the yellow checks are repeating at another frequency. So in the data, you can see you get a fundamental peak at the frequency of the stimulus, and you get harmonics of each of these stimuli. And what you can also see is you get an interaction harmonic, which is identifying when there is a population of neurons that are responding to both of the stimuli. So this can be quite an interesting measure to manipulate and see at what point you don't have the same population of neurons processing both stimuli. So my research is looking at low-level color perception. Now, the human visual color perception system is based on comparing the signals from 3 types of cones, the S, M, and L cone. And there are suggestions that because the S cone is relatively outnumbered by the M and L cones, that the scarcity of S cones means that the signal might be amplified by the visual system. So if the visual system is able to amplify any consistently weaker signals, is it possible that with colorblind individuals where the red-green discrimination is consistently diminished, is the visual system able to amplify these? So here's a brief introduction to anomalous color vision. So here with individuals with full color vision, the red-green discrimination is going to be produced by the difference at any point in the spectrum between the responsiveness of one cone with the responsiveness of another cone. So here with the normal observer, the difference at this point in the spectrum is quite high, resulting in a strong signal. However, with a deuteranomalous observer, someone who's mildly colorblind, the M cone is replaced by an L prime cone, which responds in a very similar way to the L cone. Now, this similarity results in a weak signal, which reduces the discrimination for color difference. Now, you can represent color vision in these McLeod-Boynton spaces, which construct a color space out of the 2 dimensions of color processing, the LM variation along the x-axis, and the S-cone variation along the y-axis. So there have been suggestions found in other research that the diminished red-green signal for the anomalous observer might be amplified by the visual system. So Boehm et al. Used maximum difference scaling, which is a process where you just collect difference ratings for pairs of colors in order to reconstruct an internal color space for how different all colors appear to each other. So here you can see when you do this study with normal observers, you get a color wheel replicated. But with colorblind observers, you would expect that the red-green axis would be squished, where because their discrimination is reduced, the colors would appear more similar to each other, and therefore you'd get more of an oblong MDS solution. However, when the studies run with colorblind observers, the MDS solution shows red-green discrimination that's much more like normal observers. So this is suggesting that even though the signal is reduced at the retina, that possibly there is some gain control amplifying the signal once it reaches the V1. So we are hoping to use SSVEPs to identify differences in the scaling of the amplitudes between the retina and the V1. So by comparing the amplitudes at the retina and the cortex, we can identify differences in the scaling. So on the left, we have a stimuli that is isolating the S-cone activity by flickering between blue and green. And on the right, you can see the stimulus, which is going to be flickering between red and green to isolate the LM activity, which should be diminished for the anomalous observer. So because we perhaps expect there to be some amplification of the S-cone activity for all observers, for the normal observer, there'll be a difference between the ratio of these at the retina, but not at the cortex. And then for the colorblind observer, perhaps there will be a diminished signal at the retina for both, and then an amplification of both at the cortex. So here we have the two stimuli. So you can see the blue and green stimuli is isolating the S-cone axis along the Y here, and the red-green flickering is isolating the anomalous red-green discrimination. Now we flickered our stimuli at 6.25 hertz. So in our data, we're going to be looking for peaks exactly at 6.25 hertz. And we recorded simultaneously from the retina and the cortex. So we conducted this experiment with an EEG cap, replacing two of the cap's electrodes with the skin electrodes usually used to identify HEOG and VEOG eye movement artifacts, and instead placed these just underneath the pupil on the lower eyelid to record directly from the retina. Will these work? No, they won't. So these are our very preliminary results, as we only have data from one color deficient observer so far. Now, if we look on the left at the color normal observer, we see at the retina, the LM and the S-cone stimulation are very equal and primarily happening at the fundamental 6.25 hertz rather than the harmonic. And once this reaches the cortex, the LM cone activation is slightly greater than the S-cone activation. However, at the harmonic, we see the S-cone activation is slightly greater. So then if we look at the color deficient observer, we can see that, as we expect at the retina, the reduced red-green discrimination, the LM cone stimulation, is reduced in comparison to the S-cone. But the LM cone stimulation is greater, the amplitude to the LM cone stimulation is greater at the cortex. We do see an interesting effect that the LM stimulation is entirely creating an amplitude at the fundamental and not the harmonic. And for the S-cone stimulation, we see all of the amplitude at the harmonic and not at the fundamental. So this is ongoing research. We hope to collect more data after the pandemic and to see if this pattern continues with further observers. Yes. And in conclusion, the main takeaways from this is that the SSVEP method can produce incredible signal-to-noise ratios after very short runs of data collection, that it can be used for a number of low and higher level processes, and also that it's possible to collect steady state ERGs from the retina as well as the cortex. And I welcome any questions. Thank you.
Thank you very much, Lucy. That was fascinating.
Thank you.
Does anybody have any questions? Oh, by the way, I should just mention, if you want to play your videos, you just turn your mouse pointer off, I think is
Oh, no. Well, can I quickly go back and show people some stimuli?
Yeah.
I think it's...
Yeah, it's the disables clicking, I think.
Okay.
There you go.
Yes, so basically the stimulus is flashing fast, and the exact frequency that this is flashing is driving the amplitude at this frequency in the data. So thank you.
Okay, so it looks like Chris has a question. Chris. Hiya.
Hello.
Hello, let me just stick my video on if I can. Yeah, so I mean, it's a fairly obvious question, but given the findings that you've got, and I know that they're preliminary and only in one person, but
yeah,
do you have an explanation for someone who is not particularly up to speed on color processing to explain what might be going on there if you were to see that played out in people?
I think one take home is that we don't see the effects that I was sort of expecting with the S-cones. So the scarcity of the S-cones isn't coming out in the amplitude of the SSVEPs. So it's possible that even though there are a few S-cones, they don't need amplifying because they are amplified already by the ganglion architecture that's comparing the S-cones to the L-cones and M-cones, which is already happening at the retina, so will be recorded by our retinal ERGs. But for the anomalous observer, our colorblind observer, we do see an amplification of the reduced red-green discrimination. And whether this is happening more at the fundamental, more at the harmonic, could be saying something about the inherent processing speed of that process. So there is some talk about the magnocellular and parvocellular pathways through the LGN, that one is a faster stream of processing than another. So if a process is naturally much faster than 6.25 hertz, it might naturally just respond with more ease at the harmonic, the 12.5 hertz. So I'm not entirely sure how to interpret this, but we do see some evidence of gain control on the diminished signal, and that these are diverging into slower and faster processing speeds. Thank you.
We have a question from Jamie. Jamie asks, I'm guessing the different scale on the graphs isn't relevant, e.g. different conductances of electrodes for different people. Is that
Yeah, absolutely. So we're comparing difference in scale between the stimuli rather than comparing the scale between the retina and the cortex, because they're naturally going to be very different kind of ecosystems of electricity.
Yeah, I kind of meant the fact that one goes, well, hang on. You know, maybe they are on different scales. Yeah, I was kind of looking at it that the numbers seem to be rather different from one person to another, rather than the cortex.
Yeah.
And it's probably just something like having a big hairdo, but I was wondering if there was more interesting reasons than that. But it's.
Yeah, I'm not entirely sure. I mean, there could be numerous reasons for a difference in the scale of the amplitude.
Yeah, so the retina on the left goes up to about 2, and the other one goes up to about, you know, somewhat lower. I mean, yeah.
Yeah, I mean, one difficulty we found collecting ERGs with surface electrodes was enormous variability between observers. And of course, you've got a different physiology going on. I mean, some people have incredibly droopy, baggy eyelids. Some people we could get incredibly strong signals. Other people we couldn't pick up a retinal signal at all.
Are there any more questions? Okay, I think maybe that's everything. So thank you again, Lucy, for a wonderful talk.
Thank you very much.
Thank you everybody for coming, and I hope to see you all again next year for some more exciting CNI talks.
Great. Thank you. Oh, dear.
Nice job.
Thank you. Oh, I tore through that. I hope I didn't miss out too much content.
Yeah, it's a shame about the mouse pointer. I noticed it like a little bit too late after you'd skipped off from the slide, and then I thought, oh, I won't interrupt. But
oh no, yeah. Oh well, I'll know from now on. Okay.
One piece of feedback that I think may be useful for you is when you're referring to V1, V2, V3, V4, don't say the V1, V2.
Yeah.
It's
that's V1.
Just V1, V2, V3, V4. It obviously doesn't matter at all, but it's probably what you want to use, the language you want to, the phrasing you want to use is not there.
Yeah. Cool. I'll remember that one.
And then, other than that, it was really good.
Yay. Thanks. Okay. Well, bye.
Enjoy the sun.
Enjoy the sun. Yeah, absolutely. Bye.
See you later.